Hello Biology 1 student. This lesson will focus on Competency for Biological Organization, or the cell. I have included your performance level descriptors for informational purposes only. This lesson is designed to review the Biology 1 content you learned on Competency 4 in your Biology 1 classroom. In this lesson, you will differentiate among cell types. You will describe the functions and structures of major cell organelles. You will describe the events of the cell cycle. You will compare and contrast mitosis and meiosis. And you will describe and identify examples of levels of biological organization. Here you will find your assessment verbs and scientific terms associated with Competency 4. Before we get started, I would like for you to download and complete the Foursquare vocabulary sheet for assessment verbs and scientific terms that are unfamiliar to you. Let's get started with the review process. Cells fall into two major categories. Eukaryotes are cells that enclose their DNA in nuclei. Prokaryotes are cells that do not enclose their DNA in nuclei. Here you will find a picture of a prokaryotic cell, which is a bacterium. Notice that the genetic material floats freely in the cytoplasm. It is not enclosed in a nuclear membrane. Here you will find a eukaryotic picture of an animal cell. Notice the nucleus. Here's a picture of a eukaryotic plant cell. Again, note the location of the nucleus. The eukaryotic cell can be divided into two major parts, the nucleus and the cytoplasm. We have discussed the nucleus on the previous slide. The cytoplasm is the fluid portion of the cell outside the nucleus. Many cellular structures act as if they are specialized organs. These structures are called organelles. They are literally little organs because they perform specific functions for the cell. The nucleus is the control center of the cell. The nucleus contains nearly all of the cell's DNA. The nucleus contains the nucleolus. The assembly of ribosomes begins in the nucleolus. The nucleus and the nucleolus have been labeled in the diagram of the cell below. The mitochondria are the powerhouse of the cell because they supply the cell with energy. Your cell will also contain a structure called the endoplasmic reticulum. It is the complex network of transport channels. There are two main types of ER, endoplasmic reticulum. You have your rough ER and your smooth ER. Rough endoplasmic reticulum have ribosomes attached to the surface, and it is the site of protein synthesis, as you can see in the diagram. Notice the ribosomes attached to the rough endoplasmic reticulum. In the smooth endoplasmic reticulum, ribosomes are not attached to the surface. It is the site of lipid synthesis and plays a major role in the detoxification of drugs and toxins in the cell. Ribosomes are small particles of RNA and protein found throughout the cytoplasm in prokaryotic and eukaryotic cells. There are two major types of ribosomes. Attached ribosomes make proteins that are transported out of the cell. Free ribosomes make proteins that are used within the cell. The Golgi body, sometimes called Golgi apparatus, modifies, sort, and package proteins into sacs called vesicles. The vesicles separate materials from the rest of the cytoplasm and transport these materials from place to place within the cell. Next, you have your lysosomes. Lysosomes are small organelles filled with enzymes that function as the cell's cleanup crew. They defend the cell from viruses and bacteria. They also break down damaged or worn out cells parts. Sometimes they're referred to as the garbage disposals of the cell. Your vacuoles are sac-like membranes, and they serve as storage structures in the cell. In plant cells, there's a single large central vacuole filled with fluid. Plant cells' vacuoles are larger than animal cells because they must store the products of photosynthesis. Cells have a cytoskeleton. The cytoskeleton gives the cells their shape and provides support for the cell. There are two major types of protein filaments that make up the cytoskeleton of the cell. Microfilaments support and help move the cells, and microtubules are important in cell division or mitosis. And here you will find a picture of these structures. Centrioles play a major role during mitosis. Some cells contain specialized structures for locomotion. Examples of these cells include cilia, flagella, and pseudopods. Cilia are projections from cell surfaces that aid in locomotion and feeding. Cilia are also used to sweep substances along surfaces. Cilia are found in animal cells, protists, and prokaryotes. Flagella are projections that aid in locomotion and feeding. Flagella are found in some animal cells, prokaryotes, and some plant cells. Pseudopods are temporary cytoplasmic projections that aid in the movement of protists. Chloroplasts. Chloroplasts are organelles that capture light energy and convert it to chemical energy through a process called photosynthesis. Plants and some other organisms contain chloroplasts. Chloroplasts have sacs called thylakoids, which contain a green pigment called chlorophyll. Chlorophyll absorbs light and plays a key role in photosynthesis. Here you will find pictures of chloroplast. Here's a practice activity. Identify the cell structure by the function that is listed in the left column. Here are your answers. The cytoprotein synthesis, ribosomes, the clear fluid inside of the cell, cytoplasm, produce usable forms of energy for the cell, mitochondria, digest excess or worn out cell parts, lysosome, manages cell functions in eukaryotic cell, the nucleus, provides temporary storage of food, enzyme, and waste products, vacuoles, the site of lipid synthesis, smooth ER, the site of photosynthesis, chloroplast, firm and protective structure in plants, cell wall. Let's continue. The cell membrane surrounds plant and animal cells. It provides supports and also protects the cell. Most biological membranes are selectively permeable, meaning that some substances can pass across them and others cannot. The composition of nearly all cell membranes is a double-layered sheet called a phospholipid bilayer, which gives cell membranes a flexible structure and forms a strong barrier between the cells and its surrounding. And you see a picture of that in the diagram at the bottom. The head groups of lipids in a bilayer are exposed to water, while the fatty acid tails form an oily layer inside the membrane from which water is excluded. Each phospholipid has a hydrophobic and hydrophilic end, as you can see in the diagrams. Because the proteins embedded in the lipid bilayer can move around and float among the lipids, and because so many different kinds of molecules make up the cell membrane, scientists describe the cell membrane as a fluid mosaic model. Cell walls surround plants, algae, fungi, and many prokaryotic cells. The main function of the cell wall is to provide support and protection for the cell. The cell wall lies outside the cell membrane. Here's a picture of a cell wall in a plant cell as well as a prokaryotic cell. Let's compare plant and animal cells. Notice the structure in the first column. If that structure is present in an animal cell or plant cell, it will be indicated by yes. If it's not present or found, it would be indicated simply by no. Note the lysosomes. It is found in animal cells, but it's very rare in plant cells. Here's a practice activity. Is this a plant or an animal cell? If you said plant cell, you are correct. Plant cells are usually green and contain chloroplasts. Practice activity. Is this a plant or an animal cell? This is an animal cell. It has no cell wall or chloroplast. Substances are moving in and out of the cell at all times, and there are two methods of cellular transport. Passive transport is the movement of materials across the cell membrane without using energy. Active transport is the movement of materials against a concentration gradient using energy. There are several types of passive transport. We're going to look at diffusion. Diffusion is the process by which particles move from an area of high concentration to an area of lower concentration. Note the example listed in the slide. Here we will place a drop of food coloring in a beaker of water. The molecules of the food coloring move from areas of high concentration to lower concentrations in the water, eventually reaching a state of equilibrium, as you can see in beaker 3. Facilitated diffusion uses transport proteins to move other ions and small molecules across the cell membrane. This process does not require any additional use of the cell's energy. It is still a form of passive transport. Osmosis, another type of passive transport, is the diffusion of water through a selectively permeable membrane. In the diagram, you would notice the sugar molecules on the left and look at the concentration of the water molecule. So you have more water molecules located on the right side of the beaker. So osmosis takes place. The water molecules will move from the right side of the beaker to the left side of the beaker because of concentration. Let's look at the effect of osmosis on cells. If the cell is placed in an isotonic solution, this is a solution where the concentration of solutes is the same inside and outside of the cell. Water molecules move equally in both directions. And if you can see in the picture, the animal cell and the plant cell, you have water molecules moving at the same rate in both directions. Nothing really happened to the cell or the size of the cell. In a hypertonic solution, the solution has a higher solute concentration than the cell. A net movement of water molecules move out of the cell. Again, moving from areas of higher to lower concentrations. So therefore, the water molecules will move out of the cell and the cell will decrease in size. In a hypotonic solution, the solution has a lower solute concentration than the cell. A net movement of water molecules into the cell causes the cells to swell. Therefore, the cell will increase in size. Let's look at active transport. Remember, active transport requires energy in the form of ATP to support the process. Endocytosis is the process of taking materials into the cell by means of enfolding the cell membrane. Exocytosis is the process of releasing large amount of materials from the cell. The diagrams at the bottom provide additional information on endocytosis and exocytosis. Let's practice. In what type of solution would these cells be found? Remember to use your knowledge of osmosis. This is an isotonic solution. This is a hypertonic solution. And the last one is a hypotonic solution. In a hypertonic solution, remember the cell will decrease in size. In a hypotonic solution, remember the cells will increase in size because of the movement of water. Let's practice. What process accounts for the changes in lab setup A? In lab setup B, structure Z prevents the movement of sugar molecules into side 1. Which part of the living cell serves the same purpose as structure Z? Take a moment to answer that question. What process accounts for changes in lab setup? A. The process of diffusion allows molecules to move from higher to lower concentration and reach a state of equilibrium. Which part of the living cell serves the same purpose as structure? Z. The selectively permeable membrane of a cell controls the movement of molecules in and out of the cell. Let's look at the levels of biological organization. Atoms and molecules make up cells. A group of cells working together will form tissues. A group of tissues working together will form organs. A group of organs working together will form systems, and a group of systems will form organisms. Cell division is a process to prevent the cell from becoming too large. It also produces new cells. Cells reproduce by a cycle of growing and dividing called the cell cycle. The eukaryotic cell cycle consists of 4 phases: G1, S, G2, and M. Interphase, the time between cell division. It is a period of growth that consists of the G1, S, and G2 phases. It is not a part of mitosis. M phase, the period of cell division. Here's the diagram showing you the entire cell cycle. Let's briefly look at what goes on in the cell cycle. In G1, the cell is going to increase in size and produce new proteins and organelles. In the S phase, DNA is replicated or synthesized. In G2, you have more growth and production of organelles and molecules that are required for cell division. In the M phase, the cell nucleus divides through mitosis, and the cytoplasm divides through cytokinesis. Let's briefly look at the stages of mitosis, starting with prophase. In prophase, the nucleus disappears, the DNA condenses, the chromatids are joined by a centromere, thread-like spindle fibers appear, and the centrioles move to the opposite poles or opposite ends of the cell. And here's a picture of prophase. In metaphase, the main thing that is going to happen, you will notice that the chromosomes will line up in the center or the middle of the cell. In anaphase, the chromosomes separate and move in opposite direction. In telophase, the chromosomes arrive at the poles. The chromosomes begin to uncoil and become less condensed. The cell begins to separate. 2 new nuclei begin to reappear. The spindle fibers break apart and disappear. Cytokinesis is division of the cytoplasm. The process is different in animals and plant cells. In animal cells, the membrane is drawn in until the cytoplasm is pinched into 2 equal parts. In plant cells, a cell plate will form between the 2 divided nuclei, and it will divide the cell into 2 separate cells. This slide shows cytokinesis in animal and plant cells. In the animal cell on the left, the cell membrane is drawn inward until the cytoplasm is pinched into 2 nearly equal parts. In plants, the cell membrane is not flexible enough to draw inward because of the rigid cell wall. A structure known as the cell plate forms halfway between the divided nuclei. The cell plate gradually develops into cell membranes that separate the 2 new cells. This chart compares mitosis and meiosis. Mitosis is a form of asexual reproduction, and meiosis is a form of sexual reproduction. In mitosis, there is only 1 division, and in meiosis, there are 2 divisions. Mitosis occurs in body or somatic cells. Meiosis occurs in sex cells or gametes. Mitosis produces 2 diploid cells, and meiosis produces 4 haploid cells. Mitosis play a role in growth and repair. Meiosis is responsible for the production of gametes and provides genetic variations or differences. Here's another diagram comparing mitosis and meiosis. Let's practice. Identify the stages of mitosis. Figure A is in metaphase because the chromosomes are lined up in the center of the cell. Figure B is in anaphase because the chromosomes are moving to opposite ends of the cell. While viewing a slide of rapidly moving animal sperm cells, a student concludes that these cells require a large amount of energy to maintain their activity. The organelles that most directly provide this energy are known as. Let's see what we can eliminate as answer choices. It is an animal cell, therefore we know that we can eliminate C, chloroplast. The student concludes that the cell requires a large amount of energy. What organelles are associated with energy? We know that it is the mitochondria, the powerhouse of the cell, so D is the correct answer. The diagram below represents movement of a large molecule across a membrane. Which process is best represented in this diagram? A, active transport, B, diffusion, C, osmosis, D, passive transport. We know that ATP is the energy compound of the cell. This indicates that the process requires an energy source. Diffusion and osmosis are examples of passive transport. Energy is not required for these processes. Active transport requires an energy source. Based on this information, we can eliminate B, C, and D. A is the correct answer. The main difference between prokaryotic and eukaryotic cells is A, eukaryotic cells have nuclei. B, prokaryotic cells are always much larger. C, prokaryotic cells do not have a plasma membrane. D, eukaryotic cells are always much smaller. Based on simple recall content, we know that the correct answer is A. Which of these statements is supported by data shown in the table? Let's take a moment to look at the table. Now let's look at our answer choices. Eukaryotic cells vary in covering and in food production. All right, we see changes. Vary means changes, so we see that there are some changes in food production as well as the covering. Each of the kingdoms has different organelles for metabolism. That is not true because if you look at metabolism, most of the kingdoms show the mitochondria. So we can eliminate B. All cells have nuclei for control of cell functions. We see that monomer does not have nucleus for control of cell function, so we can eliminate C. Most kingdoms are made up of prokaryotic cells. We know that prokaryotic cells contain nuclei. Again, we can eliminate D based on that information, so the correct answer is A. The eukaryotic organisms described above should be classified as: it has a cell wall, it has chloroplast, so that should automatically tell us that it is a plant cell, B. Some unicellular organisms are motile and have the ability to move. Some are non-motile, Which cellular structures are associated with movement? A. Chloroplasts, B. Ribosomes, C. Vacuoles, D. Flagella. Let's review some of our answer choices. We know that chloroplasts play a role in photosynthesis, so we can eliminate A. Ribosomes play a role in protein synthesis, so we can eliminate B. Vacuoles are storage organelles, so we can eliminate C. The correct answer is D. Flagella. Which phase of mitosis would be seen next? A. Picture A, B. Picture B, C. Picture C, D. Picture D. The top picture shows chromosomes lined up in the center of the cell. The cell is in metaphase. The next phase involves chromosomes moving to opposite poles or sides of the cell. This is anaphase. C is the correct answer because this is a cell that is in anaphase. Anaphase follows metaphase. What is the correct sequence of plant cell mitosis? We know the chromosomes are thread-like fibers at the beginning of mitosis. So we know the C is probably going to be our first slide. In B, we show the membranes are disappearing. The nuclear membrane begins to disappear, and we see the chromosomes have become condensed, so therefore B is our next sequence. The chromosomes are moving to opposite ends of the cell, so that's A, and you see the cell plate forming in the center of the cell, indicating cytokinesis is about to occur, so that is in telophase or D. So the correct answer is B, C, D, A.